talk, I delivered that we have used these molecular tools to develop a neo-isogenic lines and evaluate real economic effect of some alleles that control plant height. And we did find that there was no economic benefit coming from these alleles. So now, as I said before, we are breeding back. So what we are doing, if we did in the previous study, we took the short alleles from UK variety and increased the, Kazakh variety and increased the plant height of the UK variety. Now, using our molecular diagnostic markers, we are breeding back. So what we are doing, we are taking short alleles from the UK wheat and delivering precisely to the Kazakh wheat in order to make it a little bit shorter and optimize its plant height. Now, with these available tools in my lab, which is led by my PhD student Jules, we have obtained already F1 near-isogenic lines. So they still have a lot of genetic background from the Kazakh line, but you can see, even though they are first affiliate, you can see a clear height difference. It is not statistically significant, but almost 0.06. So when we do again one more backcross, I'm sure we will get a significant difference in between the heights of the neo pairs. So when I was doing my PhD, we thought why those alleles, although it's a plant height, were not nobody was able to pick up them, identify them. So I was thinking that Central Asian wheat, specifically Kazakh wheat, might have a founder effect. So basically, breeders were breeding for specific adaptation alleles that drive the maximum yield in that environment, and they were crossing local varieties with locals, or either Russian or Ukrainian or Central Asian varieties that are genetically very similar. So when we crossed the Kazakh variety with the UK variety, we had a chance to we had an access to those exotic alleles. So then I asked breeders, what's your favorite variety from different breeding stations? So we collected 5 varieties and crossed with Paragon again. So now we are generating more for mapping populations. This is the first my research project when I became independent researcher with my new group, and we won a small grant funding to do a postdoc. And at that time, Simon at the John Innes Center, he was doing a whole genome sequencing for Watkins. And fortunately, when we were about to genotype the population, new microarray came out, which consisted of 43,000 markers that came from the whole genome sequencing of Watkins collection. At that time, it wasn't available for everyone, with some discussions with Simon and the local distributors from Thermo Fisher, we were able to obtain that microarray. From 40,000 markers, there was not a lot polymorphic markers. At around 10,000, 12,000 of them were useful in order to create a genetic map. And this work is led by my BSc student, Madi. And what I'm saying is here, so even though it had consisted of 10,000 markers, it was very useful just to conduct a pairwise analysis between the genotypes. So how they are genetically similar. You can see the KZ Ranispelaya, which means Kazakhstanskaya Ranispelaya. It is up to almost 90% similar to Selernaya jubileinaya based on the genetic information. And the most distinct lines is the Paragon and the Prakharovka. So 70 something percent. So we can trust this genetic data. Using this, as I during my PhD, as we were not able to identify a genetic control of plant height in Central Asian wheat, then one of the main aims of creating these mapping populations was that we should have some access to those regions or in the genome that control plant height in Kazakh wheat, because in Kazakhstan it's a very droughty area. We have to sow wheat deeper in order to reach the moisture. And I was thinking breeders breed for tall varieties because of the longer coleoptile, as Johan mentioned. But it wasn't the case when we compared the coleoptile length with the Paragon and with Pamici Esiva. And here we found that out of four, two varieties only have a significant coleoptile length difference compared to Paragon, Prakharovka and the Telena Yubileyna. You can see down here, we mapped some QTL that are coming from the Kazakh varieties, which increase the coleoptile length. So now we developed some diagnostic CAS markers, and they are working very well. This is a very new results recently, basically 1 week ago, and we did some haplotype analyses. As expected, you can see green bit is the Kazakh genotype, which increases the coleoptile length. The coral bit is the Paragon bit, which has a shorter coleoptile length. We couldn't see any statistical significance between the coleoptile length of A and B alleles. I think this is due to low sample size of the coral bed. So if we increase the sample size of this population and test these markers on a bigger populations, I think we can get that significant difference between two alleles. So we are working on that now. So this bit is over. In my previous talk, I mentioned about CAWBIN, which stands for Central Asian Wheat Breeding Panel, which was created in 2018. There was a big meeting between UK and the all Central Asian wheat breeding community. And as a result of that meeting, we created a Central Asian wheat breeding panel, which in short we call CAWBIN, that consisted of around 450 lines. During my PhD, we genetically standardized those 450 lines using a single seed descent method. And Keith group at Bristol, they were quite generous, and they are genetically fingerprinted these population with a 35K axiom microarray. In Kazakhstan, Yermekbayev, he did a GWAS on winter types. In Uzbekistan, Alexey Margunov, he phenotyped this population in several locations in Uzbekistan. At the moment, I have a project that we are also doing a large-scale phenotyping within Kazakhstan and beyond Kazakhstan, and also we are trying to have these at least half of this population fully sequenced with 10x with the colleagues from China. So it will be a huge data. Currently we have aims, but we don't know. Basically, the main aim of sequencing, we want to see genetic history of breeding in Kazakhstan, also Central Asia, how it is shifted, how the genetic landscape of the variety is shifted over the time. In the next coming slides, I'll be talking about CAWBIN and the PCA plots, PCA analysis that came from the genotyping with a 35K axiom. As you can see, there is some subclusters, and the one on the top is far from the rest. So then we expect that, okay, there is a kind of interesting things going. So it's not very genetically close. They might be, some of them might be close, but it is interesting for deep and sophisticated genetic studies, which can be seen from the 3D PCA plot. So on the top subcluster, we don't know we didn't know that what was the things. In order to address these questions, we analyzed Calvin in combination with Jerry-Flax and Watkins. Of course, the merging genetic maps is kind of technically challenging, but if you have the right tools and the right understanding about the map, it would be very useful. So we analyzed them together. As you can see from the PCA plot, that green bed is the Watkins collection. Watkins, as you know, it's a as important as the Vavilov collection. That one is the blue bit is the Jerry-Flax, Western European lines, and the orange bit is the Central Asian wheat panel, which is Calvin. So we can see some overlapping lines. That was interesting for us to zoom in those regions. So if you look, what is interesting for me was, if you this PCA plot resembles kind of flipped global map showing the genetic history of wheat breeding coming from Fertile Crescent and just slowly distributing. So, these things go towards Central Asia, that thing goes towards Europe. So when we zoomed out the overlapping region between Calvin and Watkins, there was interesting lines. Most of them were USSR lines. Why? Because we know that there was a scientific collaboration between Vavilov and Watkins, and we expect that Vavilov himself deposited these lines. So then, in order to see the whole picture, we went, we merged the maps. As you can see, Western European lines, Jerry-Flax, they clustered together. UK varieties inside it. The rest is, spreading towards Watkins collection. And I fully agree with Brenda that 60, how many percent of politics? 80%. Okay, 80%, the politics plays a role significantly, because when you look at the German varieties, you can see FRG and the GRD. FRG being close to European lines and the GRD being close to USSR lines. So this genetic data is very important for us. Also, there was when you look at the Central Asian collection itself, there was 4 subclusters. First one is the in the down, so CA1, Central Asian 1. It is a spring genotypes, Kazakh and the Russian. Second one is the winter genotypes from Central Asia. Third one is the Afghani lines. They were, when I grew them in the UK in a glass house, they were very tall and like a land races. So they are close to the Watkins lines. Also, there was a CA4 which is overlapped with Watkins lines. When I looked at them, they were Timofeevi and the Milaniti synthetics or integration lines which were developed in Kazakhstan. Then, just for the sake of curiosity, I just pulled out only Kazakhstani lines, and you can see from the PCA plot that we have 2 separate breeding scales. First one is the spring wheat, second one is the winter wheat. So when you look at when you zoom out this region, uh, we got even more clusters. For example, we have some facultative lines. Those facultative lines clustered two subgroups, one being close to winter genotypes, second one being close to spring genotypes. So from now, I'll be talking about the importance of speed now. So again, Brenda introduced speed breeding, and this method was first emerged in Australia, and Brenda was pioneered bringing this technology to the UK when I was doing PhD, and the protocol was quite available. We wanted to have this technique in Kazakhstan, in Central Asia as well. Since that time, that protocol was expanded on winter wheat and recently on rice, but it's called speed flour. There is an importance of speed breeding. So, of course, you have many ways of getting your specific allele or changing mutation with many ways. For example, Cas9, genome editing, transgenetics, many things. But speed breeding, with the speed breeding, you can be as successful as those methodologies. So also the Randa introduced the importance of the light. In speed breeding, there are two important wavelengths. That is, one is the green and blue. Each has their own effect on plant growth and development. So then we were, because the bringing technology to some environment is easy, but making it work, adapting the protocol is another thing. So we wanted to start a small prototype. So we call this one is a speed breeding, but garage style speed breeding. Although we spent a lot of money on it, because when the technology comes from developed countries to developing countries, their prices sometimes increase by tenfold. So if you spend $2,000 in the US or UK or in any other European countries, we spend sometimes 20 or 30,000 USD in order to have this garage-style simple speed breeding facility. In that facility, we were quite successful optimizing our protocol, because protocol coming from Australia and the John Innes Center, we expected they might work and they might not work. Now we have our next generation speed breeding facility, and then we are doing some we are still adjusting our protocol. For example, it's a simple, you can hang the lamp, but what should be the distance? For example, John Innes Center says it should be 220. Australian colleagues, they say 150. So we tried both, and then you can see that this is the difference. Up to early booting or booting stage, you do not notice any phenotypic difference. But after that one, you can see a clear difference in development, also in yield capacity. And we tried using several varieties, and we can easily generalize that 1.5 meter was the best solution for local genotypes to be grown under speed breeding conditions. Of course, a lot of challenges which we didn't know at all, because you might think, okay, this is a phosphorus deficiency. You can find some photos that showing this is a nitrogen deficiency, but when you put them, they don't work basically. So we had a lot of challenges under our speed breeding. For example, this one. Nitrogen deficiency. phosphorus deficiency and the deficiency of other microelements in our speed reading conditions. So their cause was we had to change the potting mixture. If you are living in the developed countries, you can just order them. At Johnny Center, when I was working, we just ordered them. Okay, I'm growing wheat. Just give me the suitable potting mixture. I want to grow wheat and rice. We have to solve all of these problems by ourselves. So this was some pictures from the working process, and we developed a 17 treatments to grow wheat and to come up with a final solution, which says glucose should be the main base, the perlite 80%, sand 12%, biohumus 20%. Now this is the best solution at the moment to grow wheat specifically in our speed breeding facility. So we tested our potting mixture in bigger pots and smaller trays. It should work in both cases. As you can see, clearly the difference is clearly visible. On the left-hand side, the plants are feeling not well. On the right-hand side, they are growing happily. So this is the yield difference in yield. After that point, okay, we have the right potting mixture. Now we have to manage soil nutrient availability. So basically, how much of NPK we do need in 1 liter pot or 2 liter pots. So I did. He's the boss of speed breeding in my lab, and he was doing a lot of experiments. I'm having the same problem like previous speaker. Okay. Treating plants with once a week, once 10 days, once in 20 days, and they came up with the best solution you can see from the picture, which is in the middle. What was the solution? I think. Sorry, I forgot. Once in 3 weeks, provided these nutrients. On the left-hand side in the circle, the plants that were untreated. So the rest is the comparison against that control. So now it's a beautiful picture of our next generation's speed breeding. In my team, I didn't have any plant physiologist. Recently, I joined. She graduated from Turkey, and now we are trying to understand the effect of exogenous treatments on yield in general, on plant height, on health under speed breeding conditions. So this is Aybek, who is the boss of speed breeding, as I said before. We are doing some kind of mutational genomics. When I say mutational genomics, we mutagenized with EMS, the Pamitia, which I used for my PhD because it has a whole genome sequencing and exome capture. I think we were not successful having our mutant population because there are some 1,000 lines. We just genotyped them with some past markers, which we have in our luggage, and we couldn't see any segregation. So then I assumed maybe the mutagenesis didn't work. So Brenda, if you could help us with the protocol, that would be great. Thank you. So yeah. Also, this is the result of the 3 year of my research, and we are not holding all these results, and we are very happy to share our experience, even if it is limited, and capacity building. For that, we every year organize workshops. When I joined Jetsu University in first year, we got a scholarship or grant from BBSRC with Christopher Wally and Anna Bacchus and Lucy Wingen, and we collected emerging young scientists across the Central Asia, explaining the power of
molecular breeding in crops. Also, last year we received some funding from my own grants and the IOFS, which is the Islamic Organization for Food Security, under the OIC. Perhaps this money is coming from King Abdullah University or Saudi Arabia. And we run also some interesting workshop on speed breeding. And what we do, we try to convince traditional breeders. It's not easy, I know, but we are trying to do so. You can see from the picture that she seems happy. She's a traditional breeder. She seems happy. So she didn't believe us at the beginning. Yeah. So this is my small team. We are trying to focus in separate areas. We are not the best ones, but we are trying. Although we are separate, we are heavily interconnected, and we discuss, we chat a lot every day. So yeah, that's all. Thank you very much for your attention.
Thanks so much.
Yeah.
This wonderful achievement. That's how wonderful you establish.
Do you think so?
Yeah.
Okay, thank you.
Yeah, especially the greenhouse for the speaker. It's so wonderful. Yeah. Are there any questions now? The topic is open for discussion. Yeah.
Yes, ma'am. Please, just simple question.
Thank you, Mana.